Welcome to today's Tech Talk, brought to you by the Monash Partners Comprehensive Cancer Consortium. My name is Associate Professor Toon Nguyen Dumont, and I'll be your host for today's Tech Talk. I would like to begin by paying our respects to the traditional custodians of the lands from which we join together today, and acknowledge their elders past, present, and emerging, as well as any members of the Torres Strait Islander and Aboriginal community who may be among us today. So the Tech Talks aim to provide an informal and interactive forum for our professional research community to discover and discuss the latest cancer research technologies. And we're really keen to hear about how they can be applied in a practical way to support our research. So the Tech Talk format offers plenty of time for Q&A, and we really do encourage you to ask your questions at the end. Maybe share your experience, some key learnings and tips and tricks, as well as any potential challenges you've encountered using the tech we're going to discuss. And today, I'm very pleased to welcome Angela von Czarnecki. Angela joined SomaLogic in 2023 as part of the Global Scientific Engagement team and has close to 15 years of experience in utilizing high-throughput proteomics and genomics methodologies. She specialized in both reproductive health and oncology and has supported customers in both research and clinical settings to achieve their goals. And in her current role, she's helping to bring awareness to this next generation of high-throughput proteomic technologies and help customers design their studies to generate new insights into their disease of interest. So for the presentation, could I please ask everyone to please mute your microphones and turn off your video to minimize disruptions. So thank you, Angela, and I'll hand over to you.
Thank you too for the lovely introduction, and thanks for having us for your MPCCC Tech Talk. It's a pleasure to be here. So yes, I'm here to talk to you about our technology that's enabling high throughput proteomics. I've titled it the next proteomics in oncology. Apologies for the slightly clickbaity title. But yeah, we will be talking about SomaLogic and the SomaScan assay, how it works, and how that can be applied with a couple of oncology case studies, and hopefully have time for Q&A as well. So just a little bit of background on the company. We were founded way back in 2000, but have only been publicly traded since 2021. So there's a lot of research and development that went on before we had our first commercial assay. We started with a 1,000 protein panel and have continued to increase on that till today and into the future as well. That's our main goal in providing access to as many proteins as we can with our technology. The company's headquartered in Boulder, and we have over 1,000 issued and pending patents, over 900 peer-reviewed publications, and over 3 billion protein measurements made to date. So definitely more work that's been done in the US, but trying to bring this technology out globally as well and put it in the hands of researchers. And just a little bit about how our company is structured. There's two ways to receive data. We have a fee-for-service model where you send your samples to Boulder, Colorado, and then we also have authorized sites which are placed around the world. So for those interested to bring their technology in-house, they can do that. So our main differentiator in the world of proteomics is what we call our menu size, or the number of proteins that we can target in plasma and serum. Up until late last year, that was over 7,000 protein measurements. And now with the release of the 11K, that's over 11,000 protein measurements, which is far outpacing the other providers, especially for binding assays like we have. And just a few key features, we're able to target 10 logs of dynamic range of protein concentrations in blood and serum, plasma and serum. So that's proteins that are usually around that femtomolar range to the micromolar range. It's fairly conservative in the amount of sample that you need. So 55 microliters of serum or plasma is what's run on the robot. And it's very highly reproducible signal. So pretty consistently, median CVs of around 5% for the 7K. And we're still validating the 11K and generating those metrics. And the core of our technology and the way that we're able to target so many proteins in solution in a reproducible fashion is our SomaMer reagents. So we use single-stranded DNA, which they're called aptamers. So it's single-stranded DNA that has a 3D shape. And since it has a 3D shape, then it's able to bind in that lock and key kind of fashion with proteins and recognize them very specifically. So down to a specific epitope on a protein. And you can see in this sort of X-ray crystallography graphic, we have a target protein in the white gray color. Our single-stranded aptamer is the teal color. And what's specific to our technology is the modifications or the side chains that we've added to the DNA backbone. And these modifications aid in binding affinity, so increase the binding affinity of our reagents. They increase just the number of different molecules that we have in our library that we can then select a protein against. And those modifications really interact with the nooks and crannies in the protein and sort of help that to bind quite exquisitely to the target. So you might be familiar with other proteomics technologies, say antibodies. Using DNA has a couple of advantages over antibodies in that they're chemically synthesized, so the defined DNA sequences are the same every single time. Makes it very highly reproducible with really high batch-to-batch consistency. And we have this, these modifications that enable this really large library of molecules for us to put through our development when we're adding new proteins to our menu. Makes it very scalable. So the process of adding a SomaMer reagent to our menu and adding a protein target to our menu starts with the SELECT process, which stands for Systematic Evolution of Ligands by Exponential Expression. Bit of a mouthful, but it's the process by which we add a protein to the menu. And we don't actually design aptamers for a protein. We actually combine our library of modified oligos with a target protein, allow the ones with the highest affinity to bind. Then we'll wash away anything that's unbound, elute the bound oligos, the bound aptamers into solution, amplify those up and use those for the next round of selects. And this sort of continues, and we can include different challenges to select for different properties as well. So apart from high affinity, we're also selecting for a slow off rate. So once a SomaMer is bound to its protein target, we want it to stay bound for a long period of time because that's another feature that we use in our assay to increase the specificity. And I'll show you a bit more about how we utilize that when we go through the steps of the assay. But from the selects process, we end up with a short list of aptamers. We sequence them so we can see any motifs that are, say, bead binding motifs and exclude those and put the rest through primary validation, which will include a plasma dilution. So our reagents are fluorescent. We can measure the fluorescence from the amount of reagent that we're getting from the assay, carrying through the assay. And if we dilute the plasma with a serial dilution, we want to see that signal going down in a linear fashion. We also sort of do the opposite of that, where we spike the target protein into plasma and buffer so that we can see that we have a nice long linear range of quantification if we end up adding that to our menu. And a combination of that data goes into assessing the dynamic range, how much dynamic range this individual aptamer will have above and below what is estimated to be endogenous levels in a normal healthy population. So we have cutoffs for those. So the aptamers that make it through will then go to the affinity selection and work out the binding constant. And then we have cutoffs for binding constants. So from there, we'll choose the best aptamer and put that through some confirmation as well. So every aptamer receives confirmation via pull down and SDS page gel. So we'll pull down the target or the protein with our aptamer. We'll run that against our starting protein and make sure they're running the same. We can also do that with nearest neighbor, so very highly homologous proteins to make sure that we're getting limited or no binding for very closely related proteins from our aptamer reagents. And we publish this information on our menu of proteins on our website, menu.somalogic.com. And we are working to get as many of those aptamers confirmed with mass spec as well, and their protein targets, which again, we add to this website, the menu. And apart from mass spec confirmation, we also have performed ourselves and collected from published information any other orthogonal confirmations that there happen to be to confirm the specificity of our reagents. So for the 7K platform, over 70% of our reagents have some form of additional orthogonal confirmation, and some even have 2 or 3 forms. And that could be in the form of correlation with an antibody assay, with transcriptomics, or say a cis-pQTL, which is generated from DNA studies, from GWAS studies. So highly validated content that we're targeting. So into the assay itself, and one of the challenges of proteomics assays is the very wide range of concentrations of proteins that you get in plasma and serum. It's a bit of a dumping ground for everything that's coming from cell lysis and circulating around in that matrix. And you have some very high abundance proteins like your albumins and transferrins, and also very low abundance proteins like your interleukins and cytokines, which traditionally has been very difficult to target all of these proteins in the same assay. The way that we structure our assay to still be able to measure all these proteins at once is by just splitting the sample into 3 different dilutions. So we have a 1 in 5 dilution of plasma or serum that you're typically targeting proteins that are in the femtomolar to picomolar ranges, and that's where most of our content is. So around 85% of our Soma reagents go into with that 1 to 5 plasma dilution or serum dilution. And then we have a 1 in 200 for your nanomolar concentrations, and your 1 in 20,000 for the very highly abundant proteins in the micromolar ranges. And that's the dilution. That's actually all the sample prep that's involved before we start the assay. There's no depletion of proteins, there's no protein extraction. So all we're doing is diluting that plasma or serum before we introduce it to the Soma reagents. So yeah, the first step of the assay itself is the binding step, where at this stage the Soma reagents are bound, they're immobilized on magnetic beads. And I didn't go through earlier the functional groups that are attached to each Soma that utilized in the assay. So we have a Cy3 fluorophore, and we have a floater cleavage linker and a biotin group, so that helps with the immobilization. And we run the binding step for 3.5 hours so that the Somas can bind their cognate proteins, and we'll get specific complexes formed at this stage, a little bit of non-specific binding as well. We do some stringent washing, so anything that's sort of lightly bound is likely to get disrupted with these washes, and a little bit of increase in specificity here with our specifically bound complexes. Still a little bit of non-specific binding, but we'll address that in the next steps as well. And these complexes, so everything that's unbound is washed away, then we'll tag the complexes with biotin on the protein end as well, and wash the rest of that reagent away. The next step is our photo cleavage and kinetic challenge. So we expose the samples to UV light, just literally put the plates under UV light on the robot, and that will break this photo cleavage linker so that these complexes are then in solution. And we run this kinetic challenge for, I think it's around 20 minutes. And because we have selected our Soma reagents to have slow off rates, they're more likely to stay bound during this process. So they're not going to be falling off. They're going to stay very tightly bound to their protein partners. And non-specific complexes, they have a very fast off rate of sort of seconds to minutes. So they're going to be disassociating. And then we have a competitor molecule. It's called a polyanionic competitor, and that's going to take up any binding surfaces of the proteins to prevent rebinding. So this is another way that we increase the specificity of the assay. So after the kinetic challenge, we need to recapture our specific complexes, and we do that by the biotin group on the protein and wash away any, say, somomers that have come unbound from non-specific complexes, any extra competitor molecule. We do still catch the proteins that were involved in non-specific complexes, but because the somomer has been washed away, there's no way to get any signal from this protein. So that's essentially dark. And we have our specific complexes captured on these beads. Then we'll elute the somomers, we'll sort of denature slightly the proteins so that the somomers are released into solution. And that eluate is what we will use to hybridize to a microarray. And in this case, I think maybe in any case, but the microarray is really just a substrate for us to bind our SOMA reagents to, so that we can read the fluorescence, which is actually coming directly from the SOMA itself. So because we're using single-stranded DNA, we can have the complementary strand of each of our SOMAs present on a microarray. So they will bind, and then we can read the fluorescence. And through this process, we've translated the relative abundance of a single epitope on each protein to the relative abundance of somomers, and then in the readout, it's relative fluorescence units that we're detecting from these somomers. So your data that you receive is a list of all the somomers and all your samples and relative fluorescence units for each of those reagents. Just quickly on the addressable sample types that we have with our assay, I've talked about plasma and serum. We also can process urine and cerebrospinal fluid, and all four of those, we have matrix match controls. So if you're running cerebrospinal fluid, we run matched cerebrospinal fluid controls on every plate. They're not to act as a control group for your experimental comparisons. They're to participate in the normalization and the quality control of the assay. But it's quite nice that we have those matrix match controls. We do have a pretty long list of other samples that you can run with the assay. We call these our non-core samples. So they don't have the matrix match controls, and depending on the sample type, you're going to get different levels of signaling from the assay. And we can also support non-human samples as well. So your mouse, rat, dog, and guinea pig, we have the most experience in. And the level of signaling that you're going to get is usually related to how homologous those genes and proteins are to the human proteins as well. So we've done some experiments to show how much is signaling with those four animals that I mentioned, but we do have pretty wide compatibility. Depends on your risk tolerance as well, if you'd like to do a pilot study and see how it goes. Okay, so a couple of case studies. How am I doing for time? Good. And they're from pretty recent publications from last year. And one of them is more on the sort of early research side of things, and one of them is more on the clinical end. So I wanted to show two different examples for the ways in which our technology can be used in oncology. First one being the plasma proteome of growing tumors. So this was work that was done with the University of Colorado and actually colleagues of mine within SomaLogic as well. And the study goal was around early detection of tumors. Obviously, early detection is the goal when it comes to oncology. And wouldn't it be very nice if we could have, say, a blood test that could be a routine blood test that could pick up the presence of tumors? So the question they're asking is, what is the first tumor-associated proteins that can be detected in the blood as those initial cells establish residence in the tissues of origin? And can we use those proteins to create a model to predict the presence and identity of those tumors? So a lofty goal. And this was work that was done in mouse xenografts. So they had human cancer cell lines. So 2 different cell lines from each of these 4 different tumor types: lung, breast, colon, and ovarian. And these were injected subcutaneously into the mice, and tumors were allowed to grow, and monitored the size of those tumors, and then used the SomaScan 5K, so an earlier version of the assay with 5,000 protein measurements, to monitor those protein changes in these xenograft mice. And I'll take you through in more detail some of the findings, but some high-level highlights were the presence of 15 blood-based biomarkers that were consistently differentially expressed independent of the tumor type. So they were common amongst all of those different tumor types. And also the identification of sentinel proteins that could be used in a pan-cancer prediction model, and able to predict the presence of any tumor with a limitation of greater than 3.9 cubic millimeters in size. So the first tumor type that they started with was the non-small cell lung cancer cell lines that were injected to the mice, and there was 2 chosen: one that was treatment sensitive, the H1650, and one that was treatment resistant, H1975. So they were implanted subcutaneously on study day 0. And here on the left, I'm hoping you can see my cursor, because this might be confusing of the amount of data I've got on the coming slides if you can't. But
Yes,
so
we can.
Thank you too. All right, so they monitored the tumor volume over the course of the study. And you can see the 1650 is a little bit slower growing than the 1975. And at day 0, they collected blood. They also collected 4 to 5 additional time points, depending on, it was based on the size of the tumor, not the exact day. So there was a little bit difference in days between the 2 different cell lines. But the blood that they collected was processed into plasma and run on the SomaScan 5K, so that they could assess which proteins are differentially abundant between that day 0, and then in the 1650 it was day 9, day 19, day 30, and day 40. And these are some volcano plots of the results. The points, each point is a protein, and the points highlighted in red are those that are greater than 2-fold change between day 0 and the given day, and also reaching significance of 0.05 or less. So you can see there's quite a lot of changes in those early days of tumor development. And they did also run control mice as well that received a sham injection to make sure that these changes weren't just happening due to the time course. So what they ended up finding was a different number of differentially expressed proteins in the 2 cell lines. When you compared them, there was actually 98 that were shared between the 2 when you removed proteins that were changed due to time in the control mice. So they found those 98 differentially abundant proteins shared between the 2 cell lines of the non-small cell lung cancer xenograft mice. And with that information, they took that into generating a model. So they used the 98 proteins as input into the elastic net linear regression model, which was regressing on tumor volume. So this model could then be applied to sort of naive plasma samples where you don't have the actual tumor volume, and it can predict the tumor volume just based on the proteins alone. So the 98 proteins, they were the input to the model generation. 19 proteins were found in this common model that shared between the 2 cell lines, with strong correlation that predicts the actual tumor volume. So yeah, it predicts the tumor volume. You can see the results here of the predicted tumor volume on the y-axis versus the actual measured tumor volume on the x. And so this is the correlation, strong correlation that you can see in the training. So this is the testing set here is the set that was not involved in generating the model. And still you can see the strong correlation. So the input to this was just the entire 5,000 proteins for each mice. and then looking at only those 19 proteins that are in the model, they can predict the tumor volume with high correlation. And this model was sensitive to treatment. So they did initiate treatment for some of the mice, and as expected, the treatment-resistant tumors in the lighter blue didn't actually have a very different growth course than those that were untreated. But the alatinib-sensitive tumors, when treatment was started, the tumor volume really rapidly declined. And so that's the results that you're seeing over here and why they cluster so much towards a lower tumor volume, which is good to see that it is sensitive to that treatment. So from the lung cancer results, then they did the same or similar experiments with the other 3 tumor types, so the ovarian, colon, and breast, and found many differentially abundant proteins, and some of those that were unique to each of those cancer types and some of those that were shared between those cancer types. So initially with the shared proteins, they went about developing a model to detect any tumor type. And you can see the results here, similar to the ones on the previous page, with your predicted tumor volume versus actual tumor volume and the high correlation there. And the other thing that they did was have a look at the proteins that were unique to each cancer to see if they could develop a model that identified the specific cancer type as well. So this is the results down here of the sort of individual models and the results with sensitivity and specificity for the 4 different cancer types, and as well able to identify any cancer type versus control as well. So yeah, using the biomarkers that were shared among both all the cell lines for each tumor type led to a model that was able to predict tumor volumes which were only as small as 0.03 millimeters cubed for ovarian cancer, 3.4 millimeters cubed for breast cancer, and 15 millimeters cubed for colon tumors. So it does depend on the tumor type as to what's the lower limit of the size you can detect. I thought it was really interesting that the size for ovarian is quite small. And of course, these need to be scaled up when you're talking about human versus mice. But yeah, with 94% specificity and 100% sensitivity. So some kind of very promising work that's been done in mouse. So yeah, mouse models of the human tumor growth showing the emergence of early protein biomarkers for tumor presence in plasma. Interesting to see shared biomarkers for all the cancer types and unique ones as well that could be used to generate these models. And yeah, the protein-based tests do have the potential to enhance the performance of liquid biopsies. Okay, second case study, and I know I'm going pretty fast, but I do want to make sure that we've got a little bit of time for questions at the end. So the previous case study was more in the discovery side and the research side. Now I wanted to give you an example that is more in the clinical side, and that is with the analytical validation of the PROPHET test, which is a commercially available test from OncoHost for the decision-making and guidance in the treatment in metastatic non-small cell lung cancer. So OncoHost is one of our customers. They have entities in Israel and North Carolina. Their lab is in North Carolina. And this test was born out of research done with SomaScan data and machine learning algorithms applied to patients, non-small cell lung cancer patients that were pre-immune checkpoint inhibitor treatment. And then they have the clinical data to generate a model that is predictive of a response to that immune checkpoint inhibitor therapy based on progression-free survival at 12 months. And the data I have to share with you today is not how that model was developed, but if you're interested, there's references on their website, and most of it is this specific paper here. But the work that was done in the research setting went on to be the grounds for their clinical trial and clinical validation. That is a prospective multi-center trial with sites in the United States, Europe, UK, and Israel. Over 40 active sites, over 1,700 patients recruited to date. And that's not the data I'm going to show you today either. But they have now launched their first commercial offering with the profit tests in non-small cell lung cancer and the predictive benefit of the PD-1, PD-L1 therapy earlier last year. And I believe they've also recruited metastatic melanoma patients into the clinical validation as well. So there may be another test coming. But this is the work that they published on their analytical validation of the profit test. And so I wanted to show you this to show how SomaScan data can hold up to the clinical validation required to offer a test to patients. So study goal is obvious, validation of the performance characteristics of the profit test. And because the profit test includes both the experimental and computational components, so the plasma profiling plus the machine learning algorithms, they've got the different assessments to assess the analytical validity of the experimental precision, experimental accuracy. They showed some cross-platform comparison as well, which I'll show, and computational precision as well. So again, I'll take you through the data, but the median intraplate and interplate CVs they found were quite low, so lower than 5%. For their model proteins, the median intersite accuracy is 88% for all proteins, and the computational precision, which was how many samples and had their results that were in complete concordance over repeated measures, was 93%. So, excuse me. Okay, so first was the experimental precision, and having a look at the variation that they were seeing. Firstly, intraplate CV, and this was done by looking at technical replicates of 4 samples times 4 replicates, and then repeated with a second technician as well. And you can see the CVs for, and these are specific to the model proteins. Anything that's in light blue is for a model protein, and the darker blue is for all proteins. But for the model proteins, hovering around that 3 to 4% with fairly low spread in the data. They plotted all of the CVs that they were getting for all of the proteins, which is in blue, and again, model proteins in light blue, to see where their model proteins sat compared to all the proteins that were being measured. And they do actually skew slightly lower CV. This is probably due to the way in which targets are selected for models and the performance standards they have to hold up to. So, but overall, still some fairly short tails going into the higher CVs, and most of it clustering around the 4 to 5%. And then for between plates or interplate CV, they ran 4 different runs over 4 different days, and actually had, still 2 different technicians preparing the samples, and showed a similar CV, so not too much different to the intraplate CV here, hovering around that 4%. And similar with the histogram of where those CVs lie compared to all proteins. And then to measure experimental accuracy, they performed a 2-site comparison. So they ran samples at the SomaLogic laboratory, which is in Boulder, Colorado, and they ran the same samples in their OncoHost laboratory in North Carolina. So sort of identical setups, the same liquid handling, the same microarray reader, and they wanted to assess of all the proteins, how many had the same RFU values being generated by those 2 different sites. And that was assessed by the RFU value being plus or minus 20% was sort of the acceptance criteria. And for all proteins, it was 88% accuracy between the 2 sites. And they do note in their paper that for their model proteins, the accuracy is higher, but they don't actually say that number. But this is just one technician. From the second technician, there's an additional wobble at the end, but I think you could see that the accuracy is quite high for those model proteins, especially that they're looking at. Okay, so for the computational precision, they were running the full assay, obtaining a profit score, which is the probability of a clinical benefit from the immune checkpoint inhibitor therapy. And the score ranges from 0 to 10, with a 5 being the cutoff to generate either profit negative or profit positive result. So they had 14 different samples and 6 replicates or 10 replicates, depending on the sample. And they wanted to see that for those repeated measures, that their algorithm is generating the same result every time. And for the most part, that's true. And 93% of the samples had 100% consistency in their result. You can see there's one sample that's sort of close to that middle, the median's close to that cutoff, where it was inconsistent. So fairly good overall. And the last thing that they did was a bit of cross-platform comparison, which is curious. I'm not sure if it adds to their analytical validity, but I wanted to show it up anyway, since it was part of their publication. And what they did was for both all proteins, but this is for their model proteins, and there's 191 proteins in their model. They had a look at the available published data and who had Spearman rank correlations for the SomaScan data versus another platform, say like immunoassays or proximity extension assays, and what rank they published and compared that to their data. They generated a data set, that compared SomaScan to proximity extension assay as well, and plotted the Spearman rank correlations here too. So you have the results for immuno, PEA, and mass spec, and overall good correlation, some lower and non-correlation items. And for what they're doing, so they generated their discovery protein set with SomaScan data. They went on to validate that and use SomaScan in their clinical trial as well, and they're using SomaScan data in their test. It doesn't matter too much whether or not that's correlating well with another technology, because even the proteins here that say are non-correlated with mass spec, A, they're not using mass spec, and B, within this setting, they've been found to be very important in predicting the clinical endpoint. So yeah, the information's there, but ultimately they're sticking with the same platform from discovery to their clinical test. And this is an example of what their test result would look like. You can see that the profit score here is 3.6, which flicks this patient to profit negative. They have treatment considerations and clinical evidence, and this is all more information that's been generated in their earlier publications in their clinical trial as well, which unfortunately we don't have time to go through it all today. But like I said, just want to show you an example of the use of SomaScan data with clinical capabilities. It's holding up to that analytical validation, and an example again of SomaScan being used from that discovery phase through the launch of a clinical assay. So in summary, we do have the largest platform available for proteomics available. So up to 11,000 proteins you can target in plasma and serum with very low CVs. Our data is able to be clinically validated, and I haven't covered it here, but we've also generated our own risk assessments on different clinical metrics as well from large studies that can be applied to your data, say heart disease risk, dementia risk. They're also sort of models that have been generated on clinical data that could be applied to your samples, and is the platform of choice for many leading institutions.